Hi there, and welcome to the Systematic Review and Map Methods course. In this session, we're going to talk about data extraction and coding. You're familiar with this schematic by now. At this point in the process, we've formulated our question, we've produced our protocol, we've searched for articles, we've screened those articles, and now we have a set of full text that we know are relevant, but we want to get descriptive information and study findings out of those full texts and into some database. So in this process, we essentially want to extract all necessary information and study findings or study results. And importantly, we want to document all activities so that what we do is transparent and verifiable. And we do that transparency so that we are repeatable. And part of this process involves checking for consistency in what data is extracted and how, but also checking for mistakes or errors that might creep in, particularly when we have complex data that we might be having to summarize in some way. And along with critical appraisal, this is one of the most time-consuming parts, so it's really important to be efficient. There's no point extracting lots of information that we're not going to use, but we don't want to keep going back to articles to extract information that we should have extracted all at one time. So think about what to extract and try to extract the really key, important, necessary information. Before we get started, I just want to take some time to think about what we mean by data. So we have a term that we use to describe descriptive information about a study, the study methods, the setting, what was measured, what outcomes were measured, and what methods we used to measure those outcomes. And that descriptive information we call metadata. It's not the study findings, but it's information about the study. It would include things like information about the study site, the studied population, the intervention in terms of its strength or duration or type. If we're talking about an intervention study, it might also be an exposure. The kind of experimental setup in terms of the duration of the study, the scale of the plots that were used, the study design, for example, whether it was a randomized control trial or a split plot, a Latin square, a paired design, before, after control and impact study, perhaps a time series. We want information about the level of replication, both in terms of spatial replication and temporal replication, so replicates over time, and also if there was any subsampling. Information about the methods used to measure the outcome, and then also some information you might not have thought of. It might be really useful to report the location of the data within the article. This is particularly important if you are reading really long reports, things like theses or other grey literature, where they might be hundreds of pages long. This means if we record the figure number or the table number, for example, or perhaps the page number, we're able to rapidly go back and find that data if we need to verify anything or perhaps look for extra information so that we don't need to read the whole thing again. It's then also useful sometimes to report comments, so that if you have a feeling about the data, or if you want to provide some rich information to your colleagues, you can report that in the comments within your data extraction spreadsheet. So here's one example of a primary study that was in a systematic review. And you can see here that there's quite a bit of rich information about the study, both in terms of its latitude and longitude, in terms of the machinery that was used. In this case, it was a review about the impacts of tillage on soil organic carbon. So we can see they looked into the impacts of a tandem disc, which is a type of primary tillage equipment. We also get some information about the field more generally, that fertilizer was being applied at 45 kilograms of phosphorus per hectare. And it even goes into more details about the depth of the tillage there. So this is the kind of information that we're looking for when we talk about metadata or descriptive information. So as I said, it's descriptive information. This information is often useful for critical appraisal, and we'll come on to critical appraisal next. But this information can really be useful in making a judgment about how valid a study was. And this metadata, this descriptive info, is sometimes all that's extracted, for example, in a systematic map. So that map might just be asking questions about what is the state of the evidence, what's been studied and how, where, by whom, with what methods, etc. So this is essentially the bread and butter of a systematic map. And then we have coding. Coding is where we give a category to a study, and this is based on an a priori set of variables. For example, we might decide that we would want to classify ploughing or tillage as no tillage, intermediate tillage, or high tillage. And so for each study, we would give it one of those three categorical variables. I'll come on to the difference between coding and metadata in more detail in a later slide. And then finally, we have full data. By this, we mean quantitative or qualitative study findings. And this typically isn't extracted for a systematic map, but this is the really important thing that's extracted within a systematic review that allows us to then synthesize across studies to see what the evidence base says as a whole about an impact or effect. Extracting full data is often complicated and might require some permutations or some summary of the data as we get it into a format that can be synthesized. So coding is where we take complex descriptive data, like a description of the ploughing implements, ploughing equipment that was being used, and we convert it into one or more of a set of a priori categories. And the benefit of coding studies is that it helps to filter and search for groups of studies very quickly. If each study has a consistent way of being coded, we can then very easily filter and just get the studies that have used or investigated no tillage, for example. And when we're designing coding, you need to carefully think about the possible categories of a factor. And think about this as being tick boxes to extract information. So when you're coding, you would want to pre-select the options that someone would be able to fill in a data extraction spreadsheet. And here's the example I gave, that within one study, the tillage practices might be coded as no-till, intermediate intensity till, or high intensity till. You can combine coding with descriptive data. So if you're finding sentences or a paragraph of description about the tillage practice, for example, you could extract that as metadata, perhaps as a quotation, or you could summarize it yourself, and then also provide a code. And that's so that you could both filter studies and find rich descriptive information without having to go back to the full study itself. And because of this, you could always extract metadata in the first stage and then go back and convert that metadata from across your studies into codes post hoc. And so this coding process could be iterative, but you want to make sure that you extract the information that you need to then create those codes fully, so you don't have to go back to the full article. And you can save considerable time if you do this during screening. Here's an example of an evidence atlas that shows a systematic map database in that grey table at the bottom. We'll come back to producing these visualizations in the visualization and synthesis session. But this is just to show that here in this table you can see a combination of metadata, so the location of the study was typed in or perhaps copied, whereas the climate zone was chosen from a selection. There's only a certain number of climate zones that were included, and that was selected from a drop-down box. So sometimes within a systematic review, you'd want to have data extraction being conducted in a two-stage process. And this first step would involve extracting metadata and performing coding. And that might allow you to then perform critical appraisal based on a spreadsheet of descriptive information about the studies. You then go on to a second step after critical appraisal, where following that, you then extract full study findings. That might be quantitative or qualitative data. And the benefit of doing this two-step approach is that if you think that you will be excluding studies at critical appraisal, you don't first extract the full study findings before you have critically appraised. But then to critically appraise in a transparent way, you'll sometimes need to extract metadata and be very transparent. So you have this first step where you extract the descriptive information. You use that for your critical appraisal step, which we'll come on to in a later session. And then you will go on for the articles that you include to extract full study findings. And this allows you to be efficient so that you're not wasting resources. So most importantly, when we're planning data extraction, it's important to spend a lot of time planning. And that's because it's time consuming. So you want to establish a data extraction form. Sometimes this is done with spreadsheets, and sometimes it's done with forms like an access form, or perhaps you might have an HTML or website interface. But essentially you have a tried and tested form that needs to be filled in a consistent way. And this ensures that all information is extracted so that people don't forget which information needs to be extracted. But it also aids transparency. So someone wanting to go in and see what information was extracted and how and check for errors can do that very clearly. And it also ensures that different people extract information in the same way. One important note is just to agree what to do where information is lacking. Do you leave it blank, or do you say not reported? If you're deciding between coding and extracting data, so you might have a variable and you're not quite sure whether you want to have coding for it, which is very filterable, or have rich descriptive information, just bear in mind that the codes are really consistent, predefined categories that allow you to filter studies easily as an end user or within the process of your review. And then extraction of data involves summarizing or quoting passages from within the review, or perhaps copying numbers, quantitative information. As a tip, it's useful if you extract a quote just to make sure you put quotation marks around it so that it's very clear that it's a quote and not your summary. And when you're choosing between codes and extraction, there is a bit of a trade-off between having rich information, which you get from extracting, and having a very usable, filterable database, which you get from codes. And that's why sometimes you might want both. But remember, if you do have both, it could be very time-consuming. So you might want to have both in some cases, but one or the other for other variables. And this is just a screenshot of a really tiny part of a data extraction spreadsheet. And it just shows just how much information you would have in a spreadsheet. This, you can see already the column on the far right is CY, so it's a huge number of columns already, and a very large number of rows. In this case, each article contained multiple different studies, and each row is a single observation. So you can see these databases get very big very quickly and very messy very quickly. And therefore it's very easy to make a mistake. So thinking about how you can make sure that people extract data consistently and carefully and transparently is a really important aspect. So remember that you might find data in tables and figures where we first might expect to look, but also within text. You might find really important information buried, quite difficult to find within the text. Even more difficult, you might find that your data is buried within supplementary information. So it's important to be able to then access that supplementary information. Sometimes it might be referred to in the text. Sometimes authors might be a little bit negligent in saying where their data is. So if data is missing, you'll have to check to see whether there was whether any supplementary files along with the original publication. But if you still can't find data, you might consider contacting authors and requesting data. In some cases this works very well, in other cases it's not very successful. And if you do that in bulk, once you've tried to extract all the data, you can then email people and ask for information in quite an efficient way. If you still can't get information from authors, sometimes you can gap fill from other publications. So if the precise study location isn't mentioned in a particular article, but you know that the authors have published other articles on the same study site, you might be able to backfill and take information from one publication that's linked and fill that in. But just be careful, because you need to be really sure that that information is correct. You should also indicate where you've gap filled, just to demonstrate to people who want to come in and verify what you've done that you're not claiming that that data was within the article. Also, when you're extracting data, just have a think about trying to be user-oriented. People can really benefit from using the spreadsheets and databases that you're creating. So perhaps you would want to provide a link to the article. You can do this very easily in Excel by creating a Google Scholar link based on the title. You could also extract the email addresses of the corresponding authors from your PDFs. It doesn't take very long. Sometimes you already have this information in your citation information that you downloaded from bibliographic databases, but it might be really useful for the user of the database to know that they have a way to access the article and contact the authors. You might also, or you will also, need to decide what each line in the database corresponds to. Is it an independent study? Is it an independent outcome within studies if each study has measured different outcomes? Typically, it would be an individual observation, but what counts as an observation to you depends on what kind of analysis you're doing and what level that is. So think carefully about what you want to do with the data, how you need to present your data, particularly if you're going to be doing something like meta-analysis, and then design your database, your data extraction spreadsheet accordingly. And from my experience, I would, rather than leave blanks where data is missing, write not stated. And that's because if you come across a blank when one of your colleagues has filled in the form, if you don't write not stated, then you might be unclear whether the blank means the data was missing or it's not stated. So try to come up with a process of what to do if you don't have information in the article. And when we're extracting quantitative data, we might not only want to extract the study findings, so the outcome that's being measured, for example, we might also be interested in effect modifiers. So there might be something like temperature or soil type that is specified within the article, and sometimes also the data, the study findings themselves, are separated according to that effect modifier. For example, if they've measured different soil types, you might not want to just combine it and then extract it to get an average. You might want to extract those different soil type study findings separately. And then if you have lots of different studies that have also looked at different soil types, you can include that effect modifier as a variable in your analysis. So just think about the variables that people are separating their data over. So here, for example, in a screenshot, we've got soil organic carbon data that's separated by soil depth. And then CT, which is conventional tillage, and NT, which is no tillage, are our two main groups of data. So one option is to take an average over soil depths, but we know that the amount of carbon varies with depth. So what we'd want to do there is extract both depth and the intervention type. They also have another specific type of conventional tillage where it's just a specific crop, it's just clover. And that might be interesting to us, but it might not be. And you need to decide whether you want to extract that kind of level of detail or just focus on the interventions that are important and the soil depth, which is an effect modifier that we've judged to be important. Sometimes we find that we've got data in figures, and sometimes it's not very easy to extract. Thankfully, there are a lot of tools that help you do that, help you extract data from figures. WebPlot Digitizer is one free option, and you can copy your figure if you take a screenshot straight into this app online. You first configure the axes to tell it that the 0 here is essentially 8, and the limit, where it crosses the x-axis is 8, and where it is the y-axis limit is 18. And then you can put dots where the center of your value is and the upper and lower confidence interval, and that will then give you 3 different values there that you can calculate the error bars. Sometimes it's really difficult, particularly where the figure is quite blocky and the level of resolution isn't very high. So you might want to be a little bit careful. We can already see on the bottom figure or bottom line there that some of those triangles are sitting fairly off to the left relative to those bars. So deciding where exactly to put the point is a little bit of a challenge. When we're extracting qualitative data for a qualitative synthesis, we need to decide first of all what counts as data, and you need a clear a priori agreement across the team on what's data. And there, for example, you might be deciding that you're looking at empirical, non-numerical data based on qualitative data collection methods. Perhaps that's interviews that describe people's experience. That's one definition of data. Then you need to decide what to extract. Are you extracting quotations of the people who were interviewed, or are you extracting themes from within those quotations? So someone might give a quotation that's several sentences long, or you might be extracting themes that, for example, relate to their experience, that relate to negative feelings of a particular factor, and you might extract that as strong negative feelings as a theme. You also then need to decide whether you're extracting first or second order interpretations, so whether it's the people's interpretations of an experience or the primary research authors' interpretations of their quotations. Again, you are likely to find the data in texts or in tables. It might be that you've actually got a theoretical framework that is the data that you're extracting here. And you need to decide how much to extract depending on the purpose of the review, concerns about the quality of the data within it, within the primary studies, and the methods of synthesis that you're using, whether you're doing a framework synthesis or a thematic synthesis, for example, and as above, what your definition of data is. If you're interested in qualitative data extraction tool, there's a chapter in the Cochrane Handbook for healthcare systematic reviews that goes into a huge amount of detail and is really useful as a resource. So you can find that chapter online, open access. Sometimes the data that we're extracting, as I said, can be really complex. So the quantitative data that we're after might not be reported as simple numbers for the intervention and simple numbers for the control. The data might actually be raw, or it might be summarized over non-target variables. For example, there might be some aspects of microclimate that we're really not interested in across all articles in our review. And so we have to somehow kind of pull that back together and summarize it across the level that we're interested in. And we might need to have some calculations to do that. Sometimes it might be a simple average, but sometimes it might be quite complex. But whatever calculations we use, we need to be really transparent about those calculations, both so that we can be repeatable, but also so that we can go back and just double check that we got everything correct. So one example of complicated data is this. Again, it's from the Tillage Systematic Review, and you can see that there are 4 plots here, one from a different study location within the same article, but that data is really complex. And what we want to do essentially is to get this into one line per site where we have data for the intervention and a standard deviation around that intervention mean, and a mean for the control and a standard deviation. So there's a really complex process of recording the data from the plots and then averaging it and calculating a confidence interval at perhaps a standard deviation across that raw data. And we want to do that in as transparent a way as possible, because it's really easy for errors or bias to creep in as we do this. one way to do it is to save and report all our calculations. And we can do this by having a spreadsheet for each study. And we might, first of all, extract the data as it's presented, then have another place within that sheet where we calculate the effect sizes, and then copy the effect sizes from within that summary that we've made into a larger spreadsheet that has each effect size from each study for the whole evidence base, with each study or each observation as being a separate line. So you might want to copy and paste it in, or you might want to link it, but by having one spreadsheet or perhaps one tab of a very large spreadsheet, we just keep that separate so that all of those calculations are there and they can be checked and verified. And as I said, it's really important if you're doing this kind of thing to double check. It's really easy to make a tiny error, perhaps even a systematic error in a formula that then causes all of your data to be wrong. So it's really important to be transparent and keep your calculations so that you can go back and correct that without having to then redo the data extraction and summary process. As I've mentioned just now, effect sizes. An effect size in a quantitative synthesis is a statistical summary measure. It's a way of comparing our intervention or our exposure group with a comparator or control group. So for example, it might be the intervention value minus the control. But it involves a calculation. And just as with the calculations across variables that we're not interested in, or summarizing across raw data, we need to make sure that all calculations are transparent and that we can allow double checking and verification in the same way. So on the one hand, if we do summary across variables that aren't important to us, we need to keep those calculations. And then when we take the data and calculate a single effect size for the intervention versus the control, we also need to be transparent there as well. Consistency checking is just as important in data extraction as it is with screening. So it's important to check for errors, and it's important to check for consistency amongst reviewers to make sure that if there is a choice of what data to extract in a paper, people are extracting the right or the most appropriate data across time and across people. And the way that we do this is the same as with screening. You take a subset of studies and you get 2 or more reviewers to check for agreements or disagreements in what they extract and what they focus on. This is harder to check statistically, so it's unlikely you'll be able to do a kappa statistic because it's not a binary choice of yes or no. But in essence, if 2 reviewers disagree on anything, you need to discuss it and understand why, and then improve the level of detail in the instructions in terms of what to extract. And check again. And as I said, basically, it's important to double check all calculations and ensure that the equations are correct in spreadsheets. Partly this could be double and checking for copy and paste errors, but also where you might have a mistake in a formula. And if you do any checking, it's important to report this in detail to demonstrate that you've taken necessary steps to really make sure that what you've done is robust and as systematic and reliable as possible. So just to summarize, hopefully I've shown that there's a really strong need for repeatability, reliability, and transparency in data extraction, that you should carefully plan the process before you start, and that consistency checking and double checking for errors is really important. I've shown some big scary data sets on spreadsheets, hopefully, that just show how complex it can be. So planning and carefully checking is a potentially time-consuming but really important step. And it's much less time-consuming than going back and having to redo it because you're not quite sure. I also talked about the difference between metadata extraction and full quantitative or qualitative data extraction, and we talked about metadata versus coding. And we've talked about some examples of the data that's usually extracted within the kind of studies that we deal with in environmental evidence in particular, like study context, design and methods, and the findings.